We said that biochemistry is the study of chemicals of life, and we classify those into two categories, okay? Organic and inorganic. And we're going to first talk about inorganic. But right here on the first page, what do we got pictured? Anybody recognize that? DNA. We're not going to get to that today. It's an organic compound. It's the very last one, or very last type of organic compound that we'll talk about. Okay. Let's first talk about the inorganics. So, features of inorganic compounds include those that are small. If you look at examples that are listed here, O2, HCl, CO2, and H2O, there's not much to them. A couple, 2, 3 atoms. We're going to see today and next time large organic compounds. Okay. These guys are going to lack carbon chains. That does not mean that they're going to lack altogether any carbon. No, like CO2 has an atom of carbon. But carbon is the backbone for organics. So these guys are not going to have that carbon backbone. They're going to be made up of either ionic or covalent bonds, which we know ionic bonds are between ions, positively charged cations, negatively charged anions, which is what holds HCl together. We also see like O2, CO2, and H2O, which are held together by covalent bonds. Organics are going to be primarily made up of covalent bonds. A key feature is that they cannot be used by cells to perform complicated biological functions. They're more simple in how we use them, basically, is what that states. Obviously, we know we need oxygen second to second, minute to minute. Water, we go days without it, we're in big trouble. So they're very important. We're just using them a little bit simpler than how we use the organics. And they don't have genetic material in them like DNA does. They don't provide us with muscle contraction, which is like what your proteins can do. Okay. DNA and proteins are examples of organics. Okay. O2, HCl, CO2, H2 are all examples of inorganics. Okay. Speaking of H2O, we talked about it last time. Makes up 55 to 60% of a lean adult's total body mass. Okay. So it's very, very important to us. Other types of inorganic compounds include salts, acids, and bases, which we'll talk about salt, acids, and bases here in just a bit. Let's talk about H2O. The most important and abundant of the inorganic compounds in all living systems. This applies to all things that are living. Plants and bacteria and fungi, other animals. Not just the things that live in water, everything that's alive. Without water, we would die within a matter of days. We talked about some of the special properties. On Monday, there was a table with 4 properties, description of those properties, examples of how that applies to us. Here's a list of some more, along with those 4. Okay, the first one is that it is a chemical reactant. It serves as the medium for most chemical reactions in the body. Since water is going to make up over half of our body, those chemical reactions that we talked about the other day, synthesis, decomposition, exchange, reversible, guess where they take place a lot of times? Within water. Because guess what's inside of our cells? Intracellular fluid. We also have extracellular fluid, interstitial fluid. Fluids everywhere. Those chemical reactions that are taking place within water. Very important. We also know, based on what we learned about the other day, that water participates as reactants and products in chemical reactions. You guys remember what reactants are? They're the starting materials in a chemical reaction. Products, therefore, are the ending materials. Okay. Remember we talked hydrolysis and dehydration synthesis the other day? Hydrolysis is where we use water and break apart something bigger. That's decomposition. We talked about catabolism the end of Monday. Anabolism involves dehydration synthesis, where we remove water and build something together. Okay. So water is involved actually in the chemical reactions, not just where they take place. It has a high heat capacity. We discussed this. Can absorb or release a relatively large amount of heat with only a modest change in its temperature, which helps us maintain body temp during environmental temperature changes. High heat of vaporization. We talked about this. Explains why it takes a lot of heat to turn water into a gas, boil water. And we mentioned that as water evaporates, it basically removes a large amount of heat with it, helps to cool our skin. This illustrates why dry fit and Under Armour clothing helps keep you cooler than just like a cotton t-shirt. Okay, those clothing items will wick sweat away from us, which helps it evaporate a little bit better. Okay. So that is actual science into why those things are a little bit better than just a regular old t-shirt. It's a lubricant, and that goes into cohesiveness there. We talked about that the other day. Major component of mucus and other lubricating fluids in the body, especially necessary where internal organs touch and slide over each other. It's a solvent. We discussed this the other day, okay, that it dissolves things that are placed in it, like salt as an example. Okay, our body is full of fluids that have things dissolved in them. Okay, we'll talk about that a little bit more here in just a second, forming solutions. Cohesiveness due to polarity, high surface tension due to polarity, which polarity we learned about the other day can be shown with the way that we form water molecules, and that's due to polar covalent bonds between the oxygen and the two hydrogens, where those electrons are shared unequally, which means you end up with a negative region, a positive, slightly negative, slightly positive on the water molecule, which if it comes into contact with something that is also polar, like sodium ions and chloride ions in a cluster of salts, it's going to break those ionic bonds because those ions will get surrounded by water molecules. And that illustrates how things dissociate, which we'll talk about here in a minute. Now, salt water is an example of a mixture. We discussed mixtures in our last lecture as well, so it's kind of a review. We learned that there were two principal forms of mixtures, heterogeneous and homogeneous. Of the two, I said we'd talk more about homogeneous. We don't really get into heterogeneous in this class, other than when I introduced it to you in our last lecture. Defining mixture, this is a combination of elements, compounds, physically blended together, but not bound by chemical bonds. And since we're loaded up with fluids in our body, we're going to talk about the liquid mixtures, which include solutions, colloids, and suspensions. Solutions, also a lot of times referred to as aqueous solutions, contain solutes evenly dispersed among the solvent molecules. Remember, the solvent is found in highest proportion. The solutes are the things dissolved in it. The other name for a solution was homogeneous mixture. The example of this would be salt water. Okay, like I was just saying. Because the solvent particles are very small, solution looks clear and transparent. If you put a cup of pure water on the table and a cup of salt water on the table without tasting it, would you be able to tell the difference between the two? No, they're both going to look just clear as day. Okay. Colloids are similar to suspensions, except the solutes are a little bit larger. Therefore, they scatter light when it shines through, so they look a little bit murkier, what we could describe as opaque. Okay. We only talk really about colloids kind of one time in this class, and that's when we talk about the formation of the thyroid hormones. The thyroid hormones are the two major hormones that regulate our metabolism, which we know metabolism by now is that measure of our chemical reactions in our body. So it's kind of illustrating something I will talk about with something I will talk about later. Unlike solutions in colloids, solutes in a suspension eventually will settle out, which we can illustrate down at the bottom. When you take blood, let it sit in a sample or a test tube, and eventually it'll separate into three things. Now that can be sped up by running it through a centrifuge, which is a machine that will spin it really fast. What that does is separate out the things, and it does so by its density. The denser stuff will separate on the bottom. This is where the red blood cells will go. White blood cells and platelets will go in this thin layer we call a buffy coat. And then the least dense plasma will filter out to the top. Okay. Now plasma is primarily water. It's also got things like ions dissolved in it. So you could actually, if you had separated out the plasma by itself, it's probably a solution. And probably it is a solution. Okay. So together as the whole, though, blood is a suspension. We'll talk about blood. There's literally a lecture that we spend discussing blood, and that's middle to the end of the semester. Probably closer to spring break when we discuss that. Now, I just mentioned that if you put salt in water, it's going to dissociate, which is kind of a new term. Dissociate means that those ionic bonds break apart. We showed that here. The sodium ions, the chloride ions that are bonded together really nicely break apart, and the chlorides and the sodiums become separated from one another thanks to the water. Salt does this, so do acids and bases. Let's talk acids and bases first. Acids are substances that dissociate into one or more hydrogen ions and one or more anions. Now, hydrogen ions, which are abbreviated with just H+, are basically just a proton. When I say basically, that's because that's what they are, is a proton. Think back to the other day when we talked about atomic numbers. We learned that hydrogen had atomic number of 1. Atomic number told you that it had 1 proton. In a neutral state, it would then also have how many electrons? 1. But now it has a positive symbol next to it. What's that mean for its electron? It's not there anymore, is it? So it's 1 proton, 0 electrons. There's also no neutrons in hydrogen. So what's there to it? A proton. Okay, so sometimes you'll hear ions, hydrogen ions, as being described as protons. Okay. We often call acids proton donors because of this reason. Now, we can determine the level of acidity based on the ratio between the number of hydrogen ions and the hydroxide or OH negative ions that are released. So let's look at 2 examples to illustrate what I'm talking about. H2O, if dissociated or broken down, will form 1 hydrogen ion and 1 hydroxide ion. Well, if you count them up, there's 1 and 1. It's a 1-to-1 ratio. You guys all agree with me? That's equal. There is no more of these than these, right? So water is not an acid. Anybody know what HCl is best known as? Hydrochloric acid, actually. There's a good guess. We call it hydrochloric acid because guess what? It's an acid. Hydrogen ion and a chloride ion are formed when it dissociates. Well, there's 1 hydrogen ion, but there's no hydroxides. So since it's balanced towards the hydrogen ion, we could describe hydrochloric acid as an acid. If you've taken chemistry, there's a good chance you've dealt with hydrochloric acid. It's very, very dangerous. Our body actually produces it, though, in our stomach. It makes it really acidic inside our stomach. If that hydrochloric acid was anywhere else, it would be very, very dangerous. Burn us. Not good. Our stomach is unique, and we'll learn why later in the semester. Now, on the opposite end of the scale are the bases, or alkalines, which are electrolytes that, when dissociated, shift it the other way, to balance in favor of hydroxide. And so when they dissociate, they form one or more hydroxide ions and one or more cations. Instead of proton donors, we call them proton acceptors because they remove hydrogen ions from a solution. The example here would be sodium hydroxide, NaOH, dissolves or dissociates, into sodium ion and a hydroxide ion. There are 0 hydrogen ions, 1 hydroxide, balances it the other direction. Now, water, since it doesn't balance it either way, it's 1 to 1. Let's not use balance it one way. Let's say shift it one way. It does balance it. So, is water an acid? No. Is it a base? No. Water in its pure form is neutral, which is on the pH scale 7.0. The pH scale is how we describe whether something is an acid, a base, or is neutral. pH is short for power of hydrogen. It is a logarithmic mathematical scale that you'll never have to do any calculating with, but you could. It refers to the relative amount of hydrogen ion that's in the solution. You will need to know from 0 to 14 what numbers actually mean with regard to if something is an acid, a base, or a neutral. Okay. 0 to 7, 7 to 14 is basically the way you want to break it down. Dead center is 7.0. We say that is neutral, which means it is neither an acid or a base. At this point, sodium ion or hydrogen ions and hydroxide ions are equal, as it shows there in our figure. That's what pure water is. Anything above 7.0 is a basic solution or a base. You see here in our little jar of fluid, there are a lot more hydroxides than hydrogen ions. Anything below 7.0 is an acidic solution, which you can see more hydrogen ions and hydroxides. Something that is very key to understand is the farther you get from 7, the stronger it becomes. So 14 is the absolute strongest base. 0 would be the strongest acid. So if you compared seawater to milk of magnesia, which one's the stronger base? Milk of magnesia, because it's closer to 11. Seawater is around 8.5. Table 2.4 is from your textbook, gives you the pH values of various things. You will not need to know the pH of really any of those, with one exception, and that's blood, which we always want the pH of blood to be right around 7.4. Between 7.35 and 7.45 are what we would describe as its narrow limits. That means blood is what? Is it an acid, is it a base, or is it neutral? A very weak base, but it's a base. Why blood? Well, blood is the ultimate circulating fluid. And guess what happens to the pH of other fluids if the pH of blood is off? Their pH will change. So changes in the pH of blood affect the pH of everything else. We'll talk about that right now. I'll let you guys write this out, and then I'll read through and explain. Now, on the test, I might ask you, what does a pH of 1.2 mean, or 9.3? you would have to know the pH scale, what those numbers mean. But I wouldn't ever ask you, what's the pH of gastric juice or anything like that. Looking at that table, should be very obvious that various fluids in our body are very different with regard to pH. Gastric juice is the fluid in our stomach. pH somewhere around 2. That's a lot different than pH of 7.4. Okay. So they're all going to vary differently, but they all have to be at a certain area to function correctly. The reason our stomach has a low pH is so that it can digest proteins, but our small intestine that receives food from the stomach likes the pH closer to 7. Okay. Our mouth, we hate low pHs. Okay. Same thing for our esophagus. You ever get acid reflux? How wonderful does that feel? Not good. Okay. So we have various points in our body that prefer specific pHs. They all have very, very narrow normal limits. Okay. Easy illustration is 7.35 to 7.45. That's pretty darn small where we like our blood pH. Now, does that mean it's always going to be within those windows or within that window? No. We can see situations where it drops or goes above. 7.35, if we drop below it, is known as an acidosis. Above 7.45 would be an alkalosis. Now, if I said something is 7.3, does that mean it's an acid? No, because that's still above 7, which means it's a base. So when I say acidosis, it's kind of misleading for that reason. Don't think 7.3 or 7.2 or 7.1 is an acid, because that's actually a base. It's just meaning that we're kind of trending towards acidosis. We're becoming actually acidic because there are acids being built or formed in our body. Even though strong acids and bases are continually taken into and formed in the body, as I drink this Dr. Pepper, I'm bringing in acid. Okay, Coca-Cola, very acidic. Our pH of fluids inside and outside cells is going to remain almost constant because of what we have known as buffer systems, or buffers, as we like to call them. These function to convert strong acids or bases into weak acids or bases. If you want a simpler definition of what they do, they maintain pH. How they do it is pretty cool. They convert strong acids or bases into weak acids and bases. They help to neutralize, basically. And they do so by working with the protons, those hydrogen ions, either removing or adding them. One that we'll talk about later in the semester is the carbonic acid bicarbonate buffer system, which is especially important for the blood. There are other buffer systems that we'll talk about. We're not going to talk about these till very late in the semester, though. We'll kind of take a pause and not discuss pH too much for quite a while. Anybody have a guess as to which two systems are most responsible for maintaining pH? We have 11 systems: endocrine and nervous, cardiovascular, lymphatic, reproductive, urinary, respiratory, digestive, cardiovascular.
Urinary would be one, wouldn't it?
Urinary is one. The one that shocks students the most generally is your respiratory. Guess what happens if you hold your breath and you're not exhaling CO2? CO2 combines with water and forms an acid. Not lactic acid, carbonic acid. When we blow off CO2, that helps maintain our pH. That's really helpful every second of every day, because we breathe every second of every day. How do we use the urinary system to maintain pH. Urine has acid in it. Generally, it can range up to 8, so it can be basic, but we can remove hydrogen ions and acids by way of our pee. More important in the long term, pH balance. So we will talk about those later in the semester. The title of the urinary system lecture includes acid-base balance. So, that's our second to last lecture. We'll talk about that probably in May. All right, salts are similar to acids and bases, except for when they dissociate, the ions that are formed are not hydrogen ions or hydroxides. Okay, like potassium chloride, which dissolves and dissociates into potassium and chloride. Okay. Now, acids and bases will react with one another to form salts. As far as we are concerned, salts are important in the human body to act as electrolytes, important for carrying electric currents, especially in our nervous and muscular tissues. When we get into talking about neurons and action potentials, we're going to talk all about sodium and potassium, especially chloride a little bit. Without those ions, we would not be able to send signals, and therefore we couldn't do anything. All right, let's move on now from the inorganics to the organics, the other major class of biochemicals important to us. Now, with organic compounds, we're going to be studying a lot about and looking at a lot of carbon. We've learned about carbon a little bit. Carbon is the backbone for the formation of organic compounds. Many of those carbons, as you can see down at the bottom in our figures, are attached to hydrogens, forming hydrocarbons. If you study organic chemistry, you'd actually be studying compounds containing carbon. Now, the way that we form organic compounds is through covalent bonding, the sharing of electrons. And why carbon is the backbone is because of its diversity in how it can form bonds. Remember, carbon had an atomic number of 6, which meant that it could fill up its first ring with 2, and then that left it with 4 in its outer valence layer. Now, 4 is not 8, so it is not satisfied. So it's going to seek out 4 more opportunities to satisfy its octet. And it can do so in a variety of ways. Doing 4 singles, like we showed when we made methane, a couple doubles, a single and 2 doubles, or 2 singles and a double. All sorts of different ways and all sorts of different shapes can be formed, which we can show off in our images at the bottom. We can have short chains or long chains. There can be double bonds. There can be branching. We can even form these crazy rings. Okay. Now, organic compounds are made up of polymers that are made from monomers. The prefix poly means many. Prefix mono means one. Think of it like a brick house. The organic compounds, the complex type of them, are going to be like the entire structure. Each individual brick is the building block for that structure. The bricks are the monomers. And then you can build into the whole structure, which is the polymer. So the house is the polymer. Each individual brick is the monomer. Okay. This is done through dehydration synthesis. Dehydration synthesis we learned about last time. Taking something smaller like A and B removing water from it to put it together, synthesize. That's what this figure here is showing. To break down complex organic molecules, we're performing dehydration synthesis, hydrolysis. Taking AB and forming A plus B. There's the water, breaking it apart. If you want to label those images, it might not be a bad idea. Dehydration synthesis, hydrolysis. What we talked about last time, and that applies to this today. Now, attached to the carbons and hydrogens, you're going to have what are called functional groups. These are important because they give special properties and functions to the various organic molecules. A functional group is a group or grouping of specific atoms. We can see table 2.5 from your textbook gives off a list of major functional groups found in organic molecules. This is not something that you're going to have to know for the test, but some of these we're going to talk about. Amino acids. Guess what's in an amino acid? How about amino functional groups? What do you think they call them? Amino acids. ATP. Do you guys remember what that stood for? Adenosine triphosphate. Guess what's in adenosine triphosphate? 3 phosphates. And they all are going to be unique. And you'll find these in all sorts of different things, but they give special properties to our different organic compounds. Now, before we talk about those, I want to touch again on why carbon. And I've already discussed this, but that's okay. It's important to emphasize. Why carbon is so important and versatile in the building of these organic compounds. As I mentioned, carbon has an atomic number of 6. 6 protons in its nucleus, which aren't important to us as far as this goes. But what are the 2 and then remaining 4. 6 total electrons, 4 of which are in the valence layer, thereby allowing it to complete its outer energy shell by sharing and forming up to 4 covalent bonds. This allows it to make large, complex molecules possible, with the carbon atom generally being the central point from which the other molecule branches off, or for which the molecule branches off into 4 different directions. It gives carbon covalent compatibility with many different elements. Covalent bonds link carbon atoms together in long chains that form the skeletal framework for organic molecules, and these carbon skeletons can vary in length, shape, like straight chain, branch, ring, numbers and locations of double bonds. We saw those with these images at the bottom of that previous page. There will be other elements covalently bonded to available sites, like those functional groups. And this variation in carbon skeleton will contribute to the complexity and diversity of our organic molecules. Anybody know what the 4 organic molecules are? I've already mentioned some of their names. Talked about 3 of them this morning in my exercise phys class, so if you're in that class. Proteins, carbohydrates, lipids, nucleic acids. Three of those 4 are pretty well-known substances. Carbs, lipids, proteins. What's the other name for a lipid? Fat. These are things that you get in your diet, right? We can describe those first 3 as fuel nutrients. They provide us with fuel. You hear a lot about carbs and how bad they are. People are wrong. Okay. Fats generally are associated with something that's bad. They're good for us in certain quantities. Proteins, important in our body. They do just about everything in our body. If something's happening in your body, you can pretty much assume it's a protein performing that function. Okay. And then nucleic acids, like we show right here, includes, what is this? DNA. RNA, which is kind of like DNA's cousin. And then kind of like a nucleic acid, kind of not. It's kind of a hybrid between carbs and nucleic acids, is ATP. It's the very last thing we're going to talk about in this lecture, which we won't get to until Friday. Let's first talk about carbs. Carbs are essential. Don't cut them out of your diet, please. They provide most of the energy needed for life, and they include sugars, starches, glycogen, and cellulose, along with a little bit of something else. They're what our body prefers for making ATP. Some carbs are converted to other substances which are used to build structures and to generate ATP. Other carbs will function as food reserves, something we take advantage of, like if we want to carbo load before a big race. We can reserve it for later usage. And we classify carbs into three categories: monos, dis, and polysaccharides. Mono, which once again means one, di, which means two, poly, which means many. Monosaccharides are like an individual brick. Dis are like two bricks. Polys are like the entire house, made up of mini bricks. Let's talk about the monos and dis first. Collectively, we call the mono and disaccharides the simple sugars. These are useful for energy purposes. When people say avoid carbs, this is what you should be limiting. The stuff that is found in our food and drink that makes it taste so sweet, delicious. That's found in like cookies and cakes. Now, monos are short carbon chains, 3 to 7 carbons in length. Like I said, there are individual bricks, so they are the monomers of the larger carbohydrates. Example, which is glucose. Also, fructose and galactose are listed or shown in our image at the bottom. Glucose is known as C6H12O6 because it has 6 carbons, H12 for 12 hydrogens, O6 for 6 oxygens. You could actually go through, count them all up if you wanted to. I'm not going to do that because that would take too much time. Okay. You can take 2 of these guys and build a disaccharide, like sucrose, which is formed from glucose and fructose and bonded together by dehydration synthesis. If we wanted to go back to each of those glucose and fructose molecules, we would perform hydrolysis. Okay. Sucrose is best known as table sugar. Now, if you get way too much of this in your diet, you're going to convert them into fat. That's why if you eat really poorly, you can gain a lot of weight because our body's going to convert them. Like I said, monos and dis are the simple sugars. What you should try to get in your diet are the complex carbs, which is another way to describe the polysaccharides. These are the largest carbs, may contain as many as hundreds of monosaccharides. Principal form of polysaccharide in our body is glycogen, which is formed when you take a whole bunch of glucose and you bond it together, like we can see here with this web looking like structure on the next page. Each of these little purple rings is a glucose. And it says there, so there are probably, I don't know, 30, 40 of them. Okay, that's just one glycogen. Okay. We store glycogen in our liver or skeletal muscle, or I should say, and skeletal muscles, not or, it is both. The reason is, it's like filling up your car with gas. That way, you can take that glycogen, return it back into glucose form by breaking it down. The glucose then can be used for the production of ATP. Later in the semester, when we talk about glycolysis, Krebs cycle, transition reaction, electron transport chain, we're going to learn how glucose is utilized in the making of ATP. So even though we should strive to get more of the complex polysaccharides in our diet, it's not to say that simple sugars like glucose in our body aren't going to play a big role, because they will. Other polysaccharides include starch, which should be the most abundant carb in our diet. This is the storage form of glucose in plants, so it's essentially just like glycogen, except plants don't store glycogen, they store starch. Potatoes, people know pretty well that those are starchy, so is pasta. Cellulose is a structural polysaccharide in plant cell walls. It gives a good strength. It is indigestible, which means that we can't break it down, which means it goes right back out the same way it came in. Corn is something that's really full of cellulose. If you've ever eaten corn and defecated it out, it comes out the exact same way it came in. And it's actually really good for us. Okay? It speeds up the movement of stuff through our intestines. The longer stuff lingers in our intestines, I'm talking stuff as a feces, the more bad stuff can be absorbed. Okay? And that's not good. Okay? A lot of times you'll hear people refer to cellulose as fiber. Okay? And then there is a fourth polysaccharide, which is chitin or chitin. Not really discussed at all in the book, but it is a polysaccharide that's found in bugs, actually, and other insects. It gives them their exoskeleton, it's that hard outer shell, and it's also found in fungi, so not that important to us, unless you're eating bugs or fungus. Table 2.6 is from your textbook. It just lists off the various monos, dis, and polys. Like we saw a couple slides ago, glucose, fructose, galactose. Even tells you where they're found and how we get them. When you describe your blood sugar, that's what glucose is. Blood glucose is another way to say blood sugar. Fructose is found in fruits, fructose, fruit. Deoxyribose and ribose are monosaccharides important in forming DNA and RNA. We'll talk actually a little bit about those on Friday. We mentioned sucrose is a table sugar. Lactose, you hear a lot about people being lactose intolerant. That's a disaccharide form when glucose and galactose are combined. And then, maltose, polysaccharides we just discussed. So, not like you really need to study the table, but it's there if you want to. All right, moving on. Now we have the lipids. The second principal organic compounds. That's probably known as fats, which we describe as water-insoluble organic biomolecules that are generally nonpolar, making them hydrophobic. I went ahead and left up the notes I wrote for the first class. Might not be a bad idea to write up what I have written over there. I've explained what hydrophobic means. And I did so really simply as showing a frowning smiley face, frowny face, and water next to it. which means that it does not like water. The actual translation of hydrophobic means fearing water, which is what most fats do. They avoid water because they are water insoluble. It means they won't dissolve in water. Because fats are typically non-polar. Non-polar means, like I said the other day, simply doesn't contain a charge. So if you put, as an example, oil into a glass of water and you stir it up, what's eventually going to happen to the oil? It's going to separate to the top. No matter how much you stir and how much you shake it, it's not going to dissolve. Okay. On the opposite end of the spectrum, hydrophilic means liking water. It actually seeks it out. And salt is something that is hydrophilic. And salt, we know, dissolves in water, so we can describe salt as water soluble. Will dissolve in water. Now, that's the case for salt. Most fats, though, like I said, nonpolar, hydrophobic. Typically, they make up 18 to 25% of our body mass in lean adults, the key there being lean adults. Obviously, if the person's not lean, their lipid content's probably going to be way higher than that. Lipids are composed largely of carbon, hydrogen, oxygen, with proportions of oxygen being very low. They provide us with a lot of energy storage. 1 gram of fat will store twice as much energy as a gram of polysaccharide. Okay, 9 kcals per gram for fats, only 4 per gram in carbs. Okay, over twice. That's why we can convert carbs and why we would convert carbs into fat. It's more compact in how we store it. That is the fuel than carbs. It cushions vital organs like our kidneys, insulates against heat loss, that would be the fat that's beneath our skin, formation of our cell membranes, we'll talk about phospholipids next time, formation of hormones, if you've ever heard of testosterone or estrogen. Okay, those are an example of fatty hormones. And some of our fats actually aid with digestion. There are 6 different types of fats that we're going to talk about. The first type, which are the simplest, are the fatty acids. Fatty acids are, like I said, the simplest. We use them to make the next 2 that we'll talk about. Triglycerides and phospholipids. There are 2 different types of fatty acids. We have both of them shown down here at the bottom, saturated and unsaturated. Look at the amount of hydrogens and carbons versus the amount of oxygens. Okay, in both cases. Saturated, look at the carbon-to-carbon bonds. Notice that there's only 1 dashed line between them. That 1 dashed line means that there is only a single bond existing. Okay. A saturated fatty acid is one in which all the available bonds of its hydrocarbon chain are filled, so there's always a hydrogen attached. Okay, at least as we see 2. Okay, I'm not going to worry about this one here. And then there's a 3rd at the very end. With unsaturated, like we're showing here, these 2 carbons are lacking a hydrogen because there's a double bond that exists between those 2 carbons. This is an example of a monounsaturated fatty acid, which has 1 double bond. 2 double bonds would be considered 2 or more a polyunsaturated fatty acid. Look what it does to the shape of the fatty acid when it has that double bond. It puts a kink in it. That's going to affect what we call triglycerides. And we'll go ahead and pick up with this since it is 10:50. We'll start here on Friday. You guys have any questions for me?